Thank you for allowing me to present my work today. The work that I will present is about, we were trying to understand how the level of expression of neutral sphingomyelinase 2 can affect anti-melanoma immune response and impact the response of these tumors to immunotherapy, and especially the one that is targeting PD-1. So first, so cutaneous melanoma is the 90th most common cancer worldwide, and its incidence is increasing every year. This disease originates from the uncontrolled proliferation of melanocytes following accumulation of distinct mutations that are now well characterized. And it begins by the proliferation of cells during the radial growth phase in the epidermis of the skin. And then the cells can cross the basement membrane to invade the dermis and eventually go to the lymph nodes and disseminate into distant organs to form metastasis. So when the disease is detected at an early stage, it's quite of good prognosis. When the disease is detected at late stages, metastatic stages, it is of very bad prognosis. So this is a highly metastatic disease. And in the last 10 to 20 years, the key events that led to some kind of revolutionized treatment for melanoma patients was first the discovery that these tumors are very immunogenic, and the characterization of the immune microenvironment of tumors was a key factor in the development of different treatments. The other key factor that helped for the development of treatment was the discovery of mutations in melanoma cells in the MAP kinase pathway, specifically BRAF mutation, that are very important for the development of this cancer. So as I said, two types of main treatment have revolutionized the treatment of melanoma patients: targeted therapies that are aiming at inhibiting the MAP kinase pathway through targeting BRAF mutation with BRAF inhibitors combined to MEK inhibitors. So the response rate of this kind of treatment is of about 70% in patients, but there are some relapses. The second type of treatment that are mainly given to patients with metastatic melanoma now are immune checkpoint inhibitors. So these treatments aim at awakening the immune response in melanoma patients to get rid of the tumors. So basically what happens is that for an anti-tumor immune response to be efficient, T cells have to be educated by antigen presenting cells. And this is dependent on the delivery of co-stimulation signals from antigen presenting cells like dendritic cells or macrophages to T cells. But the expression by T cells of inhibitory molecules like CTLA4 or PD-1 can lead to the inhibition of the activation of these T cells, and treatments have been developed to block these molecules and awaken T cells. So as you can see in here, there has been a lot of improvement in the treatment of patients from chemotherapy that only led to about 5% overall survival after 5 years. And from 2012, there was the development of immune checkpoint inhibitors like anti-CTLA4, and then targeted therapy like BRAF and MEK inhibitors that improved a lot the survival of these patients. However, there are still some relapses, and for now, the best option maybe for patients on the long-term treatment is about the treatment with anti-PD-1 and combination therapy between anti-PD-1 and anti-CTLA4 that allow for durable responses that goes over 5 years of responses. Still, there is some big issues with these treatments, since 40 to 60% of patients do not respond to these therapies and eventually relapse. So it is necessary to try and find ways to improve the response of patients to immunotherapy. So what about the role of the sphingolipid metabolism in cancer and response to this kind of therapies? So in the lab, we are mainly working on two main aspects of this sphingolipid metabolism. We can see the sphingolipid rheostat cancer as divided between two very simplistically, two main lipids. So the ceramide is considered as an anti-oncometabolite because its implication in growth arrestant cell death. And there is, on the other hand, sphingosine-1 phosphate that has been shown through its pro-survival effects, proliferation, angiogenesis, and inflammation processes to promote cancer progression. So to shift this balance towards anti-tumorigenic properties of the sphingolipid metabolism, one way would be to inhibit the synthesis of sphingosine-1 phosphate through inhibition of SK1. And this is a work that has been recently published in the lab, which was showing that inhibition of SK1 can lead to decreased growth of melanoma tumors in mice. And this was associated with promotion of immune responses and response to immunotherapy. The other way to tackle this sphingolipid rheostat cancer would be to try and promote the production of ceramide. And one interesting enzyme that we have been working on for quite a while now is neutral sphingomyelinase 2, which is known to be activated by different factors like cytokines, chemotherapy, but also the number of cells in the dishes is able to increase its activation. And it has been linked to cell growth arrest, cell death, and exosome budding. One very interesting fact about this enzyme is for us that the gene coding for neutral sphingomyelinase 2 is found downregulated and or mutated in different cancers like leukemia and breast cancer. So our question was, what about the role of this enzyme in melanoma progression? So this is what we tried to look at by looking at different data sets. The first was the Oncomine data set, where we found that the gene coding for neutral sphingomyelinase 2, so SMPD3, is less expressed in tumors from metastatic melanoma patients as compared to patients that have primary disease or as compared to normal skin. And when we look at the correlation between the level of expression of SMPD3 and survival in patients with metastatic melanoma from the TCGA data set, we can see that there is an increased overall survival of those patients that have high levels of expression of SMPD3, which suggests that somehow maybe decreasing the expression of SMPD3 in tumors is favoring their development. So to try and understand how nSMase2 could potentially promote or inhibit the growth of melanoma tumors, we used the B16K1 mouse melanoma model, which is expressing very low level of nSMase2 and high levels of MHC class 1, which renders these tumors quite immunogenic as what is seen in the clinic. So for this, we collaborated with Chris and Yusuf, and we generated cells that were expressing high or low level of nSMase2, with nSMase2 in here being mostly expressed at the plasma membrane. And this was associated with increased neutral sphingomyelinase activity and accumulation of ceramide. So the first thing that we have done with these cell lines was to check whether this overexpression of neutral sphingomyelinase 2 could impact the proliferation of the cells. And we were actually quite surprised to see that nSMase2 expression at a high level does not impact on the proliferation of cells in vitro. It does not either impact the formation of spheroids or culture in 3D. But what was quite interesting for us was that when you try and orthotopically inject those cells in the dermis of mice, there is a decreased growth of tumors that express nSMase2 at high level as compared to the one that express nSMase2 at low levels. And again, this is associated with an accumulation of ceramide in tumors, especially C16 and C24-1 ceramide. So the next question was, well, if this accumulation of ceramide and increase in nSMase activity is decreasing tumor growth, but it is not doing so by decreasing proliferation of the cells, what's the mechanism by which it's doing that? So what we saw was that when you look at the immune infiltrate in those tumors, there is an accumulation of immune cells, and especially CD8 cytotoxic T cells. And what was really interesting was that when we tried and looked at the growth of those tumors in CD8 knockout mice, we could not see any more the difference in terms of growth, meaning that basically the decrease in the growth of those tumors is really dependent on the accumulation of CD8 T cells in the tumors. So the next question was, is this dependent on the activity of nSMAse2? So we generated, again, cell lines that express the wild type or the catalytically inactive form of nSMAse2 and injected them to C57 black 6 mice. And we saw that only the ones that overexpressed nSMAse2 wild type had a decreased rate of growth as compared to the catalytically inactive one or the control ones. And this is again associated with an accumulation of CD8 T cells in tumors as compared to tumors that express the catalytically inactive form of the enzyme. The next thing we checked was that, okay, you have an accumulation of CD8 T cells in tumors, but are they able to target the tumor cells? And we saw that there was an increase as well with high nSMAse2 wild type expression in the proportion of CD8 T cells that can actually target the tyrosinase 2 antigen expressed by B16K1 as compared to the ones that are infiltrating tumors that express high level of the catalytically inactive one. So for now, what we could see was that, okay, a high nSMAse2 activation through ceramide production and maybe other sphingolipids can promote somehow anti-tumor immune responses in melanoma, leading to decreased melanoma growth. The next thing we were wondering was that, okay, well, when we overexpress an nSMAse2, there is a decreased growth of the tumor, but it's not preventing completely the growth of those tumors. It's only slowing them down. So one possible explanation that we thought was that when we look at the infiltrate of those tumors, we saw that in tumors that express high level of nSMAse2, you have an increased expression of the immune checkpoint PD-L1 on macrophages, on CD45 negative cells, including tumor cells, and on dendritic cells, which could be a way by which there is a downregulation, or actually maybe just an inhibition and not efficient enough immune response in these settings. So that was an argument for us to try and combine those this overexpression of nSMAse2 to anti-PD-1 therapy. And when we did that, what we saw was that when you have tumors that express low level of nSMAse2 and you treat them with anti-PD-1, you do get a decrease in the growth of tumors, but no regression. When you treat nSMAse2 high tumors with anti-PD-1, we got first a small, so that was treatment after the beginning of the growth of the tumors, and we got all the tumors that regressed very quickly after the beginning of the treatment. And this was associated with a much better survival of the mice. So that was nice to see, but we also confirmed these kind of results in two other cancer models, the murine YUM1.7 melanoma models and the 41 mammary carcinoma model, which shows that really it seems that nSMAse2 can kind of jumpstart the immune response and helping then for the efficiency of immunotherapy afterwards. So how does this combination therapy works? When we looked at the immune infiltrate in tumors, what we could see is, again, when you have an overexpression of nSMAse2, you have an increase in the content of tumors by CD8 T cells. And this is even more increased when you combine nSMAse2 expression to anti-PD-1 treatment. And these CD8 T cells are also even more potent and efficient, as seen when we are looking at the production of effectory molecules like TNF and interferon gamma by CD8 T cells in these tumors. So there is an increase in the production of interferon gamma and TNF upon an nSMase2 increased expression, and this is even more potentiated by anti-PD-1 treatment. So there is increased infiltration of tumors by CD8 T cell. They can target the tumor cells, and they can as well produce effector molecules to kind of tackle the cancer. So, but the next big question for us now is, finally, how can we link in terms of molecular mechanism the increased activity of nSMase2 to a remodeling of the immune microenvironment, because this is increased activity in tumor cells. So one of the possible explanation that we are now trying to understand is whether nSMase2 could kind of lead to differences in the production of small extracellular vesicles like exosomes, because it's been shown that nSMase2 can promote the budding of exosomes, and exosomes are very important cargoes that can modulate the immune response, especially in cancer. So the first thing that we looked at, we were trying to understand whether overexpression of nSMase2 could lead to increased production of EVs by tumor cells, but we could not see any difference in terms of SEV production, nor did we see any difference in terms of their sizes or their shape. What we could see, though, is that when we were purifying small extracellular vesicles from B16K1 that express the wild type or the catalytically inactive form of nSMase2 and put them on top of dendritic cells, which are essential antigen-presenting cells implicated in the activation of lymphocytes, we could see that when these EVs were coming from cells that express the wild type form of nSMase2, DCs had higher level of expression of genes coding for key factors implicated in T cell activation, especially for the T helper 1 response, which is essential for anti-tumor responses, like genes like IL-12, CXCL9, or CCL19. Now, what is exactly the factor in these EVs that leads to this promotion, and how do they eventually in vivo promote anti-tumor responses? This is something that we are still looking at. So these results we published last March. Basically, what we saw was that increased nSMase2 activity, in our case, is able via ceramide production and maybe other sphingolipid metabolites, promote CD8 T cell responses in tumors, which is implicated in the inhibition of melanoma growth, and also the promotion of anti-PD-1 efficacy. Now, whether this is dependent on small extracellular vesicle production and whether this is independent of this kind of pathway, we are still looking at right now. So yeah, that was all. I thank all the team and the people who did the work with us and our collaborators and fundings. Thank you.